Thank you. I want to start also by thanking the organizers for inviting me. It's my first time at this incredible institute, and I have to say I've always been really intrigued by the idea of the Center for the Unknown. I think that's the best center name that I've ever heard, so it's great to be here. Thank you. So my lab has been interested in understanding how the gut-brain axis integrates information about energy from the external and internal worlds. So for example, when an organism consumes food, you get the taste and the flavor and the perceived pleasure. So that's an external signal of energy. For example, sweetness signals sugar. And then you swallow and the food is trafficked through the gut, and eventually, as we've been hearing about, you release all of these endocrine factors like CCK or insulin. And these signals ascend to the brain and play a really important role in meal termination, okay? And drive reduction. And these signals, as you've seen, can be really powerful modulators of reward. So for example, this is an old study, but it's still my favorite showing this. So this is a study by Stephanie Fulton, and she's used brain stimulation reward in mice to index reward sensitivity. And on the left are animals that have been food restricted, and on the right are animals that have been injected with leptin, which is secreted from fat cells, and it's a signal about energy stores. And what you can see is that the curve is shifted to the left when the animal is hungry, and to the right when there's a signal about energy stores. So these signals and internal states are really shifting sensitivity to like this very fundamental reward signal. So more sensitive here, less sensitive here. But importantly, these endocrine factors are not rewarding in and of themselves. And by what I mean by that is that animals will not work to self-administer leptin or insulin. Okay, so they're important modulators, but they are not themselves reinforcing. Instead, there's a second class of signals that have been discovered in the last 10 years that actually act as primary reinforcers. And where all the signal is for reinforcement in food is actually from these subliminal signals that are primarily neural, that ascend to the brain and basically bring the value of the ingested food and form reinforcement. So that's a critical signal for reinforcement in humans. And these signals animals will work for. So for example, a great example is this paper that is quickly becoming a classic from Wenfei Han from Ivan Diarrujo's group, where they used viral vector tracing to map out this circuit that goes from the upper intestine, the endocrine cells, through the vagus nerve and onward to regulate dopamine. And what they did was they transfected these cells here, specifically receiving information from the endocrine cells in the upper intestine through the vagus. They transfected them with channel rhodopsin and then implanted a laser right over their terminals in the nucleus of the solitary tract and allowed the animal to nose poke to receive stimulation of those, basically the upper intestine cells. And this is the behavior. Ivan lent me the video. And so what you can see, this is the non-rewarded port. This port is rewarded, so the laser, which you can see there, turns on when the animal pokes, and you can see it will quite happily poke to receive stimulation from this intestinal pathway. And you can quantify the behavior, as you see here, and the animal learns really to nose poke exclusively for the active port, and you even see this in extinction. And if you turn the laser on and you record in the dorsal striatum extracellular dopamine, you can see a nice rise in extracellular dopamine when you stimulate that specific cell group, but not if you stimulate an adjacent cell group. So this is gut-induced reward, and because of the time, you're going to have to take my word for it. This pathway is lipid sensing. So this is a critical pathway by which fat is reinforcing. So fat cells bind to these, there's receptors in the endocrine cells, and this is the critical reinforcing signal for fat. But as we've heard, there's actually many macronutrients, and there's actually specific mechanisms relaying reward for different macronutrients. And today I want to focus on glucose, and starting also with an older work from Ivan's lab, again using microdialysis. And here what you're seeing is, again, dorsal striatum, extracellular dopamine. In the open circles is the rise in dopamine from baseline in animals that are looking for glucose. And the interesting thing actually is the solid circles, because these animals are also looking for glucose, but they've also been simultaneously infused with 2-deoxyglucose, which is an anti-metabolic agent. So they're doing the exact same thing. The only difference is now the cells can't use glucose for a fuel. And when you block the ability of the cells to use glucose as a fuel, you completely block the dopamine release. It's a completely blunted dopamine signal. And if you look at appetitive behavior, and in this case it's licking, you can see the animals actually stop licking, as if it's no longer reinforcing. So this suggests that there's a signal created when the glucose is being used for fuel by the cells that is really critical in driving dopamine and appetitive behavior. So Yvonne and I wanted to see if we could find evidence for this in humans, and we designed this flavor nutrient conditioning protocol where people came into the lab and they sampled a bunch of novel beverages that were sweetened with sucralose, because they're non-caloric, and they told us how much they liked them. And then with the flavor nutrient conditioning paradigm, what you're doing is you pair one flavor with a nutrient and the other one you don't. So to make this one caloric, we added maltodextrin, which is a glucose polymer, so when you consume it, it breaks down into glucose really quickly. But it has a very minimal orosensory profile, meaning that the subjects can't actually detect the presence of the maltodextrin in the solution. So we added 112.5 kilocalories to this beverage, and this one remained non-caloric, and subjects came into the lab over 4 days, and on each day they would consume one of the beverages, so always hungry and in the morning. So they have two opportunities then to associate this flavor with these calories and this flavor with the no energy. But the critical thing that we did here was to take a blood sample just before and after they consumed the beverages. This allowed us to look to see the impact of drinking this thing on plasma glucose levels, which will be our proxy measure of metabolism, because the glucose needs to be available in the blood in order to be used as a fuel. Then, of course, we want to look to see if the brain response differs to the flavor that's predicting the calories versus the flavor that's not predicting the calories. So they're in the scanner here with the little drops of liquid. Again, in here, it's all non-caloric. So these are just calorie predictive flavors now. And you want to see what areas of the brain respond more to the CS plus than the CS minus, and as a function of the changes in plasma glucose that occurred days earlier. And so when we did this, you see two very selective and strong responses. So the responses on the y-axis, this is the nucleus accumbens and this is the hypothalamus, where activation, so this food cue response, is very tightly associated with how much plasma glucose levels increased. Again, days earlier, because here there's no calories. So this is a calorie, like a food cue response, and you can see that what is driving the food cue response is how associated that food was with energy availability, glucose availability. And what's interesting, actually, even though we saw evidence for hedonic conditioning, right? So these are the liking ratings of these beverages. This is unexposed. This is at pre-test. And at post-test, the liking ratings go up for the exposed beverages compared to the control. And this one is significant. So there are changes in perception and how much people like these things. But these changes in liking have absolutely no ability to touch the signal in the nucleus accumbens or in the hypothalamus. So these areas that were very sensitive to this caloric, or to the availability of glucose in the blood, are not sensitive to changes in perception. Instead, if you look at the areas that are sensitive to a perceptual change, you get perceptual cortex. So this is actually the anterior insula. Its flavor cortex in humans is where taste and smell are combined to make flavor perception. So if you change perception, make something more liked, it's not surprising that you see a sensitivity in the flavor cortex. So what this study showed then is that there's two separate circuits, one circuit that's sensitive to perceptual but not metabolic changes, and one circuit that is sensitive to metabolic but not perceptual changes. Okay, so the next thing we wanted to do was a dose-response curve study. And you see 4 talented postdocs here. They all have their own labs now. And 4 postdocs because this was a very long study. So what we did was similar to before. We had the 0 cal and the 112.5 kilocal beverage. And then we just have a few more doses, maximum 150 kilocals, which is the equivalent of a can of Pepsi. So we sweeten them all to be similarly sweet because we want to test the prediction that more calories equals more brain conditioning to the flavor. So they're all similarly sweet, and we sweeten them with sucralose to about 75 kilocalories using sucrose, table sugar, as a reference. And then we repeated the experiment. And just imagine each participant coming in for a couple of days, for each one of those beverages over like a period of a month. So a single subject takes over a month to run. It was very time intensive. So these are the liking ratings at pretest, and the idea is to see at what dose do you see an increase in liking. And we were able to replicate the previous study. So this was a significant increase, but so was this. And so what this meant is that a lower calorie beverage was more reinforcing, became more liked than a higher calorie beverage. So the first thing we thought is this experiment didn't work because this doesn't make any sense. So then we looked to the brain data, and we modeled these brain responses to these flavors using a linear model, so more response, more calories. And when we did that, we saw absolutely nothing significant in the brain. Instead, we saw responses that looked like this. So here is bilateral midbrain, and you can see responses greater to the 112.5 predictive flavor than the 150 predictive flavor, which again doesn't make sense, but it's consistent. And because this was consistent, we thought, maybe we should follow this up. So what the data means, if indeed it is the case that it is the utilization of glucose as a fuel and some signal that's generated that's really driving reinforcement, what it means is that if you drink a bottle of 112.5 kilocalories of this flavored beverage, you should have a greater metabolic response than if you drink the higher calorie beverage. And fortunately for us, you can test this rather inexpensively in humans by doing indirect calorimetry, which just measures resting energy expenditure. So people came into the lab, this is a separate group of subjects, come into the lab over 3 days, and they just consume one of the beverages on each day, and we measure before, then they come out and they drink the beverage, and then we measure 30, 40 minutes afterwards. And when you eat something, resting energy expenditure increases, reflecting the metabolism of the nutrients that you've just consumed. And so the prediction is that we're going to see greater changes in resting energy expenditure for the 112 versus the 150 kilocal beverage. Now, I know this doesn't make sense, but this is what that data was suggesting from the prior experiment. So here you're looking at changes from baseline, and this is the 0 kilocal solution. Here's the 112.5, so this is dietary-induced thermogenesis, as we would expect. And then the question is, what happens to the 150 kilocal beverage? And, I would think it should be up here. But in fact, at no time was it ever different from the 0 kilocal beverage, which was consistent with the previous study, right? So the people are drinking the 150 kilocal beverage, but it is not being metabolized. Dietary-induced thermogenesis is blocked, which doesn't really make any sense. So the first thing that we did when we got this was to, with our limited amount of money left, run everything again, sort of as a sanity check. So we had enough to run an additional 9 subjects, and everything sort of checked out. So we saw the increase here to the 112 and not the 150. We saw response in the nucleus accumbens was graded according to the change in dietary-induced thermogenesis. We saw, again, the small increase in liking for 112, but not 150. And then when we looked at the plasma glucose levels, we saw something that was really surprising. So this data here is just this collapsed across time. So you have increased energy expenditure for 112 versus 0, but not 150 versus 0. But if you look here, you can see that the plasma levels of glucose are increasing after you drink the 150 kilocal beverage. It's just not being burned, right? It's not being used as a fuel. And fortuitously, this is really good evidence then that in order for something to be reinforcing and condition brain responses and liking, it's not enough to be tasted, swallowed, trafficked through the gut, even absorbed into the blood. It has to actually be used as energy. So that was cool because that's consistent with the animal data, but it still left us with the question as to why on earth would something like this exist. And it took a while, but we eventually figured out that by making all of the beverages similarly sweet, we had done something that evolution doesn't do. And that is, when something is sweeter, there's generally more sugar there. So sweetness should have gone like this. And so we wondered, could it be that the taste is actually modulating the metabolism? And so to test this, we recruited another group of subjects, and we had them do indirect calorimetry 4 days. So we have 2 different levels of sweetness, again using sucralose, and 2 different levels of calories using the maltodextrin, which gave us 2 beverages that are matched. So this is twice as caloric, but it's also twice as sweet. And then 2 beverages that are mismatched. So it's the exact same amount of calories, but this one's too sweet, and that one's not sweet enough. And the prediction is you should see greater dietary-induced thermogenesis here compared to here if taste is regulating the production of, is regulating the metabolism. And that's what we saw. So green is matched, orange is mismatched. It's not as clean as before, but it's the exact same amount of calories present. So the only difference is this tiny little bit of sucralose. And it's really a little bit, because it's a very high affinity ligand. And that changes how much resting energy expenditure changes. And for the 112, you see a cleaner effect. And we did that just because we'd used that dose in the first experiment. So the situation, then, is that you have metabolic signals that are generated during glucose metabolism that regulate central reinforcement circuits so that the nutritional properties of the foods can be learned. But at the same time, you have sensory signals that are associated with sweet taste that is regulating the generation of the metabolic signal. And to anyone who studies learning, this starts looking a little bit like predictions and outcomes, but very different from what we're used to talking about, because this is gut-brain prediction, so sensory prediction, and then a nutritive outcome. And then the question becomes, what happens when the prediction's wrong? Can we find evidence for sensitivity in the brain to something like a prediction error? And we can actually look at that in this experiment, because instead of modeling it linearly, we can model it using the prediction error. So here, the prediction is that it's not sweet enough, or no, sorry, too sweet, and then this one, not sweet enough. So we can take the difference between the prediction and the actual calories received and model the data. And when you do this, you see in our dopamine circuit, so midbrain, nucleus accumbens, ventral medial prefrontal cortex, that in fact, it is what is driving response, what's accounting for response in these areas, is actually whether or not there's an error. So there's no error here, but there is here. So the nucleus accumbens is not sensitive to how many calories were present with this beverage. It's sensitive to the difference between the sensory prediction and the actual nutritional outcome. So this is like a prediction error. But of course, in this experiment, they're not, in the scanner, they're not actually consuming calories. So this is actually more likely reflecting an updating of a belief, right? Updating of the value of the cue based on what would have been a prediction error. And the prediction error, of course, is quite different from how we're used to thinking about it in terms of phasic dopamine to, the juice reward, which transfers to the cue that predicts the juice. And when the juice is predicted and doesn't come, then you get the prediction error. In this kind of model, the taste is the reward, right? The unconditioned stimulus. And then that phasic response, when it's unexpected, transfers to ever earlier predictors. But what we're talking about now is taste actually also just being a predictor. And so, and the actual signal, we don't know what it looks like, right? But it's happening at some distal time. We do think it involves dopamine because you've seen in animal sensitivity to the oral and to the post-oral nutrient infusions, right? And in humans also, you see dopamine changes to both oral and post-oral phases. So this was a collaboration with the Max Planck group in Cologne, and they developed this temporal clustering technique that allowed them to look at when binding potential changes as opposed to where. So in this experiment, people were lying in the scanner, hungry, and at about 20 minutes, we started injecting them with unexpected milkshake. And during this time, we'd infused radiolabeled raclopride so we could measure binding potential change related to the D2 receptor. And when we did this, we did see, in fact, and so they're 5-minute bins, but the bin closest to the onset of the oral sensation here, you see a peak change in binding potential, and then one delayed by about 10 minutes when there was no oral stimulation occurring. So that suggests, so there's evidence then in humans like in animals, dopamine's involved in both the oral and the post-oral phase, though exactly what that dopamine signal looks like in the post-oral phase is unknown. So what we've seen then is really two pathways by which reinforcing signals can reach the brain. The high road, which is what we classically think about, that is sensitive to external energy signals like taste. And then the low road with these subliminal signals generated in this case during nutrient metabolism that ascend, and actually this is really the primary reinforcer. But of course, food choice depends upon their integration. And of course, the whole purpose of this circuit is to update the values of these things based on not how much energy is in the food, but the actual physiological utility of that act of eating in that one instance of that particular food. So it allows you to very carefully titrate the value of the food, of that particular food. And this got me thinking, you know, what are the bounds of this system? So this is all learning, but at any time our cells are using fuel, primarily fat and carbohydrate. So is there any evidence for sort of online updating of values? So does the fuel that you're using at any time influence how you value that fuel out there? So, for example, we are primarily burning fat and carbohydrate, and in humans, we can index that with respiratory quotient just by doing indirect calorimetry again. And values closer to 1 reflect burning carb. Your cells are using primarily carbohydrate. And as you go towards 0.7, you're using more fat. And so we had people go into the indirect calorimeter, and we measured their RQ. And then right after that, we gave them this auction task where that we developed, where subjects bid against the computer for the opportunity to eat these different foods. And how we had created these pictures is so that we had some that were high in carbohydrate, low in fat, others that were high in fat, low in carbohydrate, and some that were equally high in fat and carbohydrate, but they're all similarly caloric. They're similarly liked, they're similarly familiar. People actually do bid more, though, for those foods that have fat and carbohydrate. And so that suggests that these signals for different macronutrients are potentiating each other. So if you have calorie-for-calorie foods that have both, it's more reinforcing. And we replicated that in the US with different pictures and showed that it's blunted in obesity. So we had 25 healthy weight people in this study, and what you can see is that the amount that they bid for foods that have fat in it was proportional to RQ. So if you're burning more fat, more carbohydrate, you value fat more. And so this is still very much in progress, but I think it's intriguing because it does suggest like online real updating of an internal state burning calories, which is burning particular nutrient, which is related to the value of that nutrient in the external world. And so with that, I just acknowledge the various collaborators. I've introduced some along the way, and it's late in the day, so end there and take questions. Thanks.